Yeah, that's it. Okay. So thanks. In amongst all those other titles, today my title is the Kevin Strauss Sandwich. Uh, I'm Jerry Vockley in between two presentations by Kevin, which is, of course, a great honor. And Kevin's already given you one ride on the Wayback Machine. I'm going to do another, and I'm going to take you on two little side trips. One is to give you a little bit of the history of gene therapy so that you can understand where the field is today. And the other one is back to your basic biology course, your Bio 101 course, because I want you to understand some of the where we are intervening in the biology of all of this. So let me see if I can get this going forward. And my disclosures, and so the first move here is to, let's see, if I can get a laser pointer to work. Yeah. Okay. Well, actually, it's just the pointer. Okay. It's to take you back to that very first biology course you had, whether that was in high school or college, and you should have heard about something called the central dogma. And what the central dogma says is that our genetic information is stored in DNA, which is in a special compartment of the cell called the nucleus. And for that DNA, the function of that DNA to be realized, it has to be turned into proteins, which, with some exceptions, are really the worker bees in the cells. These are the things that are carrying out all of the functions of the cells, including the enzymes that are broken in inborn errors of metabolism. And to do that, there's an intermediate step. So you go from the DNA, the cell first makes a copy of that called RNA, and in fact there are two kinds of RNA. There's precursor RNA and there's messenger RNA. And the reason for that is that of all of the information encoded in the nuclear DNA, only about 3% of it ends up encoding proteins. The rest of it does something else. We first didn't even know it was there. Then we started calling it junk DNA because it didn't do anything relative to proteins that we could recognize. And now we're pretty sure it's actually all doing something, which makes much more sense from an evolutionary standpoint, because otherwise it would have just been lost as we evolved from being a slime mold to us. The move from this precursor mRNA, which is an exact copy of what is in the DNA, to messenger RNA, involves cutting out those areas that don't make a part of protein, because as it turns out, all genes essentially have intermittent pieces of protein coding regions separated by other pieces of DNA that don't involve the protein. So that process is called splicing. And what you end up with, you splice out all the stuff that isn't part of the protein, is mRNA. And so when you hear about mRNA therapy for any disease, and there is an mRNA therapy that is an experimental design for MSUD, that's what that's all about. Now you get to the protein part of things. Proteins are very complicated structures, but they start off as two dimensions. They start off as essentially a string of beads, and each amino acid is a bead. And if you take a string of beads and you dump it on your dresser, what you get is a jumble. It takes no particular structure. So for that to happen, for that string of beads to make a functional protein, now it has to be folded properly. And that folding is carried out by other proteins, and eventually you get a mature protein that can do its thing. And you can have genetic defects that impact the final ability for the cell to make the right protein to be in the right place at the right time at all of those steps. And there are medications that are being developed for many diseases, including MSUD, at all of those steps. So what we're going to talk about, what I'm going to give you an introduction to today, is gene-based therapy that is one component of the concept of precision therapy that Dr. Strauss introduced to you. And that's a very, very important term, because what it implies, what it states explicitly actually, is that the same therapy is not going to be right for everybody. So what we're going to do is talk about the history of gene therapy, where it started, where it's going, and hopefully get you to where you need to be to understand some of the more detailed stuff that Dr. Strauss is going to tell you about his very, very exciting MSUD gene therapy. So I love this quote by Lewis Carroll: begin at the beginning and go on until you come to the end, then stop. And so that's the outline for my lecture today. This is a visual of what I just said to you. Options for therapy, and when you look at what an enzyme does, it starts here, and it takes a chemical and it turns it into another chemical. That's what an enzyme is, a really, really highly specialized chemical factory. And the chemical that it starts with is called the substrate, and the chemical that it finishes up with is the product. And so, and you can do things that allow you to work at the substrate end, at the product end, leucine, or at the enzyme. And one of the ways that you can fix a broken enzyme is to give you another copy of that of the gene that makes that enzyme. And so that's what gene therapy is. Now, drug therapy is traditionally a very long, complicated, and extraordinarily expensive process. In the classical design of something that is for a common disease, hypertension or antibiotics or even a chemotherapy for cancer, what you do is you start off with some incredibly large number of chemicals, hundreds of thousands or 1 million chemicals, and you look to see if they can somehow affect something that tells you something about what you're interested in. Now that's very vague, and I say that very vaguely for a reason, and that's because most of the time what drug companies are looking at when you start off with a 1 million chemicals and you're looking for something that's going to help cure cancer, you can't actually test that directly. So you have these intermediate stages where you are looking at a surrogate marker of the function that you actually want to be measuring for your drug. And so you start off with this 1 million things, and you drop them into this bin, and you measure different things over the course of the lifetime of the development of that drug that get you closer and closer to being able to understand if that or recognize if that drug's going to do what you want it to do. Most of the time, ultimately, you have absolutely no idea what you're doing until you get it into a person. And then you have to test it on a large number of people, thousands or tens of thousands of people, to understand what it's going to do. Well, we can't do that in rare diseases. And the good news is we don't have to, because we can take most of this whole funneling effect and get rid of it, because we know exactly what we want to do. We have ways of measuring that in cells, in animal models that you'll hear with Dr. Strauss, and in people. And so we can shortcut all of that and move from a 20-year drug development timeline to, if you're really good and you're really lucky, 5 years. Now, it still takes a while to get it out into general practice, but it's a directed approach to developing something rather than an undirected approach. And so we not only have precision in how we use drugs, but we have precision in how we're developing drugs. And that's much to the benefit of getting these drugs put together. So we're going to start with some definitions here, or continue with some definitions here, because what we're going to be talking about is not protein therapy, although eventually we're making proteins to do this, but nucleic acid therapy. And nucleic acid therapy can be broken down into a handful of categories. If your gene is broken, it's not working properly, it doesn't make the protein that you need properly, you can just give it a new copy of that gene. That's called gene addition, and that's the earliest form of gene therapy. Sometimes the gene is doing something you don't want it to, and so you have to silence it. That's called gene silencing. In many evolving cases in rare diseases, you don't really need to give a whole copy of the gene. There's only one amino acid in that protein that's not working properly, so you can design ways of actually just fixing that one. It's called a codon. The three nucleic acid base that defines amino acids, and you can fix that one codon, and now you have a gene that works perfectly well. That's called gene editing. We can go to the next step in the central dogma, and which is mRNA, and we can make mRNA. Gene therapy, gene editing, gene addition are at least have the potential to be permanent cures, because they go in and they give the cell a copy of something that it can keep making more of, so that you don't have to keep treating that. mRNA therapy, by definition, is temporary. You get an infusion of mRNA once every 1 week, 2 weeks, 4 weeks, whatever the experiment for that particular mRNA says is the right interval, but eventually it gets broken down by cells and it doesn't have a way of making any more, so you start over again. You take another infusion. Now that sounds like it's pretty inconvenient, but I can tell you that we've been doing enzyme replacement therapy in another group of disorders, the lysosomal storage diseases, for 3 decades. Patients who have come in every 2 weeks of their diagnosed life to get an enzyme that replaces the one that they're missing. It doesn't work for MSUD. Well, it might, but it's probably not the best way to go, because it has lots of complications. And an mRNA therapy is really a much better approach for that, because when you inject the protein, you get an immune response from the body. It's just like any other vaccine in that regard. You inject a protein, you get an immune response. Whereas the mRNA, when it gets into the cells, gets made and it's hidden from the immune system. So that's a good thing. There are some other categories that I'm not going to talk about. We can fix things that are defects that are where that splicing process isn't working properly because the signals for the splicing aren't there, and you can work at that level to try to get rid of some of the exons that are not working properly, and you can do some other things with just trying to regulate the gene expression, sometimes upregulating another gene that can compensate, or downregulating the one that's abnormal. So let's go to gene therapy. The goal of this, as I mentioned, is to get a durable expression of something. So one and done is the terminology that you'll usually use. You'll usually hear. It may not be one and done. It may be 10 years and done. We don't know, but one and done sounds like a good fundraising line, right? We'll do that. The requirements for that is that you've got to be able to get the protein that is going to eventually be made from that gene therapy, that gene that you're either fixing, you're adding. It's got to get into the right tissue, which means that the therapeutic vector that you're using to deliver it has to get into the right tissue. It's got to be stably expressed, and you don't want it to interfere with normal function of anything else. So those are the basic requirements. Now, the history of gene therapy is interesting and to some extent tragic, actually, very tragic in one case. But it really evolved from the study of bacteria, the second most simple form of life, and the way it interacted with the most simple form of life, a virus, which is, it's kind of on the edge between a piece of DNA and life. There were attempts to do this with several diseases all the way back into the 1990s. Now, the nice thing about gene therapy is that it has focused for a long time on rare diseases, because all you got to do is fix one disease. If I can't treat hypertension with gene therapy because I don't know what the genes are that cause hypertension, but I sure do know what the genes are that cause MSUD and other inborn errors of metabolism. There is a heavy focus on in gene therapy development on cancer, but it's a relatively non-specific one. It's not going in and fixing the genes that are causing cancer. It's adjusting the cells of the immune system to be able to fight cancer better. So you'll hear of CAR T therapy. That's a different thing than we're talking about here. Now, right now there are 20 plus approved gene therapies. Most of them are for cancer, but there are a handful of them now for rare diseases. This is the timeline, so you can see newborn screening that Dr. Strauss was talking about started back in the early 60s, and really is if you're talking about trying to treat a rare disease, this is the optimum time to identify it. You might not need to treat it right away, but you want to know about it because you want to keep a baby, an individual who has a genetic disease, a rare disease, from having irreparable damage caused by that disease before you can get in and do the final treatment. So in MSUD, we use the diet, and we get you to a point where you're healthy and now able to undergo another therapy. Right now, our gene therapy is liver transplant. But we'd like to be able to do something that's less invasive. Anything that starts with step one, remove liver, to me is kind of a tough way to go. Fortunately, we have people who are so good at step two, replace the liver, that it has become the standard of care in many cases for MSUD. As we move across the earliest attempts at gene therapy, they really go back to the 90s and were on their way to probably being, gene therapy's been just around the corner for the 40 years I've been doing this, and it's just around the corner still. So why is that? Well, I'm going to introduce you to a young man named Jesse Gelsinger, and everybody in the field knows this unfortunate young man. He had a disease called OTC deficiency that leads to the inability of the liver to get rid of the ammonia that the body builds up through normal metabolism, and these patients go into coma and they die. We have some tricks that we can use to keep them alive, but not very long and not very well. So they were one of the earliest targets for liver transplant, because this could get them a urea cycle that is working and allows them to survive. Jesse had a milder form of this and was able to survive to adulthood, and he was participating in a clinical trial for a gene therapy. He was diagnosed instead of at birth at about 30 months of age, and he had multiple episodes for hospitalization for his ammonia, he was the 18th patient treated with a vector that was built off of the type of virus that causes the common cold, an adenovirus. And the other 17 patients seemed to do pretty well. Within a week of getting that therapy, he started getting extraordinarily sick, and over the course of another few days, he died. And it turned out that he had a massive immune response to that adenovirus that was carrying the OTC gene. appeared to be specific to the adeno carrying part of the therapy, this viral shell that Dr. Strauss mentioned, and it stopped the field cold. It stopped the field cold for almost 20 years. And what it's required is the development of new vectors, new ways of delivering DNA to cells that are now form the basis of gene therapy. So there are a number of ways that you can deliver DNA to tissue. The most common one is still to use a viral vector, but it's a completely different vector. Even though it has the same name, it's got adeno in the name. It's AAV, adeno-associated vector virus, and it is just a virus that happens to be a hitchhiker in the lungs. When you look at the way that adenovirus attacks lung tissue, and so it's benign in humans. It doesn't cause disease, and scientists have learned to take the guts of it out and put in the ones that we want, and make gene therapy vectors, a viral particle that can deliver a gene that is necessary. And there are some long-term complications or concerns about this that we don't know. AAV vectors we say they're life. We would like gene therapy to lifelong, but an AAV vector for DNA therapy to be lifelong, that DNA has to integrate with the DNA of the cell. Because otherwise, every time the cell divides, it's going to eventually lose the DNA if it's not actively transported from one cell to the next as the new cells divide. AAV doesn't integrate except occasionally when it does, and when it does, it integrates sort of randomly, and there are long-term concerns about whether or not that could be problematic. There's been at least one report of an AAV that integrated in a spot in the DNA that activated an oncogene, a gene that when it's activated can lead to cancer, and so we're not quite there yet. We don't know what the long-term outcome of these things and what that real risk really is, and although the AAV virus is much less immunogenic than the adenovirus was, it still causes some problems. It can still cause inflammation, and so we are learning. In fact, we're learning from liver transplant, where we have to keep the body from rejecting a whole liver, to how to suppress the immune reaction to keep it from getting out of control when you do something called AAV. So. This gives as a cartoon of the basic AAV vector, which basically just has two ends of the virus DNA that allow it to make copies of itself, and then you take everything else out and you put the gene that you want in there. It can then infect a cell because it's got the outer part of the virus, but now that DNA is sitting out in the cell, and it starts making copies of its genome as RNA, and that gives you the protein that you need. Now, we've had other steps back. In fact, there was an AAV gene therapy for a disease called myotubular myopathy, and the preclinical data looked great. There was a dog model, very impressive, and there were multiple safety studies in non-human primates, and it had all the characteristics of a potentially very successful gene therapy. They started a clinical trial. They gave the lowest dose. You always start low and you work up, and those patients did well, but they weren't having quite the clinical response that they were still symptomatic. So they escalated the dose to the next higher level, and 3 patients died. Put the field on hold for another 5 years, it probably turned out that this is a muscle-specific phenomenon, so a muscle-specific phenomenon in a muscle disease. And it probably isn't relevant in MSUD or other more basic liver suspected or centered diseases. There are other viral vectors that are designed by nature to integrate into a cell and hijack the cellular mechanisms from inside the natural chromosomes. And one of these is called a lentivector. You know one of those very, very well. It's the disease that causes HIV/AIDS. And it's so good, and we understand it so well because of our studies of AIDS over the decades, that it is now being used as a gene therapy vector. Now, we've taken out all the things that make it cause immune deficiency, as much as we did with the AAV, and it can now integrate into, you can actually target it to where you want it to integrate. By a process called homology. So the downside of it is that right now we mostly can only get decent expression in a liver, and it's still not as high as you express it in with AAV. So it's moving forward. You'll start hearing about it, but it's probably not going to be the first gene therapy that is made for MSUD. All of what we've talked about, there's another way. There are non-viral ways of delivering DNA to a cell. The problem with those is they really are only liver focused right now. So they actually might be a good bet for delivering an MSUD gene therapy, but they're still not as effective as AAV in getting into cells. So if you need high-level expression, you still got to go with AAV. If you're looking at something where you can get by with lower-level expression, you can use what's called a lipid nanoparticle. Now, all of this was, let's just get a gene in there and make it work. That's the gene addition or the gene expression version of gene therapy. The other side of the fence that I mentioned was gene editing, where you're going to go in and allow the cell to essentially repair itself. And this gets complicated, so I'm not going to take any more time because I'm going to run out of time. But you might have heard of CRISPR-Cas9 in the news. This is one way we're now sort of reverse engineering things, and we're hijacking systems that bacteria use to either repair themselves or fight off infections, and use their proteins that are based on attacking DNA and tricking it into fixing DNA. So in the case of CRISPR-Cas9, what it does is one component of that goes in and makes a cut right where you want it in the native DNA, the cellular DNA. You bring in a template for fixing the DNA that you want, and when the cell goes to fix that break, it uses the template instead of the native sequence, and you've got a repair. That's not as efficient as some systems, and there are now, as I was preparing for this talk, there are dozens of ways that are using, instead of something that cuts the DNA twice, it cuts it only once, and so it's easier to fix, and there are less mistakes, and it's more efficient. So one of those is called prime base editing. There are a whole variety of these things, and I think you'll hear a lot more of this as we go forward. Now, those of you who were at the scientific meeting yesterday, you heard about a mouse MSUD muscle gene therapy that is being developed, and what you can see here is that you're seeing a lot more. These animals are gaining a lot more weight as you give them the gene therapy. They're surviving. This is something called a Kaplan-Meier curve, and you just plot how long individual animals live. And the mice without the therapy die very quickly. Mouse without MSUD, of course, live a long time. And you can see the therapy was starting to work. These mice were living longer. Most of the mice that died early, and it was probably a combination of the techniques needed to deliver the gene therapy. There's the different AAV vectors go to different tissues at various efficiencies, and there's one called AAV8 that's very, very good at getting into liver. You'll hear about AAV9 from Dr. Strauss. And here what you can see is that if you use an AAV8 directed gene delivery for the B form of MSUD, you can get 100% survival of those animals, and you can get their leucine levels down to normal. So this is a very is very good. There are a lot of there are pluses and minuses of using these various different approaches. And I think I'm going to skip over this because I think I'm probably out of time. How much time do I have here? Am I done? Okay, so I'm gonna finish up by simply saying that having the drug isn't good enough. We have to have an infrastructure that can deliver it. And that infrastructure is usually people like me, geneticists or hepatologists, neurologists, who are delivering the gene therapy. And right now, there are precious few of us who do this and who are comfortable doing it. And the economics of delivering it when you're giving a $2 or a $3 or a $4 million drug are difficult. And so we have a long way to go in figuring out how to get this into the clinic in a way that makes economic sense, that can allow hospitals to survive. Because from the hospital standpoint, I'm telling them I'm going to do a treatment that's going to give them ultimately maybe $50,000 in reimbursement, compared to a liver transplant, which gets them $1 million. Well, when I say that to my hospital president, she says, "Well, why do you want me to let you start a gene therapy program?" Because for every patient I treat with gene therapy, I cost them $1 million. There's got to be an economic model there somewhere, and I'm going to stop there because we all have to figure this out together going forward. So I will have a break later, and we'll have question time. So thank you.